welcome to all the learners. Uh, so this is a continuous series of lectures under the LMS scheme of Government of Karnataka. So the topic of today's discussion is protein structure. So this topic will be discussing in two units, protein structure 1 and protein structure 2. So proteins are polymers. They are made up of amino acids, which are the building blocks of proteins, which are small organic molecules that contain an central carbon atom. We call it as an C alpha, linked to an amino group and an carboxyl group, a hydrogen atom and a variable component called as R group. Okay, there are over 20,000 different proteins in the body. And how do proteins have so much functional diversity based upon only 20 building blocks? That we will see. In nature or the natural, there are only 20 amino acids, and these 20 amino acids, they undergo different permutations and combinations to form polymeric structures, which we call it as proteins. And these proteins do a variety of functions in our body, and we call proteins as the workforces or the workhorses in our body or in the cell. So any function is possible only because it is a protein. So whether it may be catalysis, whether it may be the muscle contraction, or it may be a hormone. So this way, proteins perform a variety of function in our body. And these amino acids, they are linked together by a bond called as peptide bond, and they form a long polypeptide. The peptide bond is formed between the amino terminal of one amino acid and carboxyl terminal of the another amino acid. This peptide bond is a rigid and a planar bond. The linear sequence of amino acids within a protein is considered as the primary structure. Proteins were long thought to be colloids of random structures. In 1934, the crystallization of pepsin, which is a protein enzyme, in X-ray beam produced discrete diffraction patterns, and atoms are ordered. So this led to the discovery of the protein structure, and a new science emerged as protein chemistry. In 1958, first X-ray structure solved sperm whale myoglobin. No structural regularity was being observed. Today, approximately, we have 50,000 structures that have been solved, and remarkable degree of structural regularity has been observed. And these protein structures give us an idea about the active site, about the composition of the amino acids, about the shape of the protein, and lot of studies can be done with these crystallography structures. We can predict the protein shape, we can predict the uh, uh, docking, we can do a lot of thing can be done by knowing the protein structure. We can understand the nature of the protein. Okay, this protein structures are conformations. They are being classified under four subheadings, right? uh, the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. The primary structure is a linear structure of, or a linear sequence of amino acid. There is no turning, there is no twisting, rather it is a straight line, right? uh, the secondary structure, so the primary structure is something like this. It is a straight, so where you have amino acids, amino acids, amino acids joined together by uh, a peptide bond. The secondary structure is the local arrangement of a peptide backbone. So this linear structure, which was a primary structure, it undergoes some extra bonding, it undergoes some twists and turns, right? It is possible due to the, it is possible due to the uh, right, it is possible due to uh, the it is possible due to the peptide backbone, the local arrangement of the peptide backbone, and these shapes are being retained or supported by extra bonds. They are hydrogen bonds. They provide stabilization to these structures. The tertiary structure, three-dimensional arrangement of all the atoms. It is the three-dimensional arrangement of all the atoms in the peptide backbone and amino acid side chains. Remember, amino acid side chains are groups, right? And the quaternary structure, it is the spatial arrangements of the protein subunits. It is the spatial arrangement of the protein subunits. Yeah, this is how the classes of protein look like. The primary structure, so which is a linear, so this is one amino acid and this is another amino acid, this is another amino acid, this is another amino acid, and this in yellow color is an peptide bond or a peptide linkage. And this is your secondary structure. In secondary structure we have two types of conformations. One is the alpha helix. As I said you, the primary structure undergoes twisting or turning, and this structure is being stabilized by the hydrogen bonds. It looks helix like structure, and it is a alpha helix. And we have another type of conformation that is called as the beta sheet. So this beta sheet looks like this, right? So it is your beta sheet, and this is also stabilized by the hydrogen bonds. And we have the tertiary structure, and in tertiary structure you can see this is a helix, this is a alpha helix, and this is a beta sheet, right? This is a beta sheet, and these two are in turn interlinked with a hydrogen bond, and this forms the tertiary structure, right? And in quaternary structure we have two different polypeptides. We have two different polypeptides. So this is one polypeptide and this is another polypeptide. We may have two polypeptides, three polypeptides, or we may have four polypeptides, and these proteins are interlinked by the hydrogen bond to give the quaternary structure. We will see all the features of alpha helix and beta sheets in much detailed way. So this is the primary structure of this is the primary structure of this is the primary structure of protein, which is a linear structure. Here we have taken an example of insulin molecule, so which is a alpha chain. We have two chains, alpha chain, beta chains, which are primary structures, which are linear structures where amino acids are arranged and they are connected. Okay, so when we look at this structure, this is the structure of a insulin. So here this is a primary structure, amino acids where they are just linearly joined together with the help of a peptide bond. We have already seen how a peptide bond is formed between the carboxyl terminal of one amino acid and the amino group of the second amino acid. They form together a CONH bond. They form a CONH bond, and this is a peptide bond. When we come to the secondary structure, it refers to the local folding of the polypeptide chain. And here you can see this is very interesting. See, I said you that these are your peptide bonds. I can just highlight it. So this is your peptide bond. So this is the amino group of one amino acid, and we have the R group. So we have C alpha N CH, C alpha N CH. So this order is going to repeat. So this order is going to repeat in the amino acid, and always in a polypeptide you have one terminal as amino terminal and the other terminal as a carboxyl terminal. So we are starting with the amino terminal and we are ending up with the carboxyl terminal. So here you can see there is a formation of a peptide bond. So this is C double bond O NH. This is a peptide bond, and here again you have a peptide bond COONH. So this way, when we start to add on the amino acids, these amino acids, they condense to form a peptides, and then they form polypeptides, right? So we have, so this is R1 group of one amino acid number 1. R2, it is the R group of the amino acid 2, and this is the R3 is the R group of the third amino acid, right? And we this CN bond, I said you that it is uh, double bond character. It is having a slightly it is having a double bond character in amide in the peptide bond. So this amide bond is having a double bond. So it also looks like this. You can also see here how peptide bonds are being highlighted. So this is your uh, C double bond O NH. So this is your rigid planar structure what I was telling you. So this is one a peptide bond, and this is another peptide bond, and this bond is having certain limitations of rotations, and that is being proven by Ramchandran plot that we will see. I think that is not there in your curriculum, right? And it is very, very important in the protein structure determination, and to prove yeah, the tertiary structure is the arrangement of the secondary structure elements in 3 dimension, and quaternary structure describes the arrangement of a protein subunits. Linus Pauling and Robert Corey analyzed the geometry and dimensions of the peptide bonds in the crystal structures of molecules containing one or a few peptide bonds. And now this is your secondary structure. So what I was telling you is, please look at this, how this uh, alpha helix looks like. So you have your C double bond O CH NH. So this is your C double bond O. So this presence of these, you can see here the hydrogen bonds which I have shown you highlighted here. I guess you can see the hydrogen, it is of one peptide bond, and the oxygen is of another peptide bond, and in between you can see the bonding, and these hydrogen bonds, they provide the stability to the helix. Even in case of the beta sheets, you can see here this is one uh, chain of amino acids, this is another chain of amino acids. You can see here there is hydrogen bonding between the hydrogen of one amino acid and the oxygen of another amino acid. So you can see the formation of the hydrogen bonds, right? This looks like pleat like, so hence we call these as pleated structures, and this looks like a coiled structure, so hence we call it as an helix. Now let us redefine the features of alpha helix. So this is a much simplified structure. So when it is asked in the examination, you can write this structure. This is quite easy to write, and it is very easy to understand also. So the alpha helix defines the interaction of closely located amino acid chains in an amino acids. The main two types of secondary structures observed in proteins are alpha helices and beta sheets. Alpha helix is a helical structure around an axis. This is coiled in right-handed. So if you see clockwise right-handed manner, so if you just start twisting, it will always turn towards right hand rather than the left hand. Hence it is a right-handed helix. The coil, this is coiled in, there are around 3.6 residues per turn. So so when this helix is completing one full complete turn, so it will have around 3.6 amino acids. It will have around 3.6 amino acids per turn. So in while studying nucleic acids, we have tried to understand that around 10 nucleotides are present in one turn, right? So this is similar like that. Thus the rise per residue come out to be 1.5 angstrom unit. So when one amino acid is turning, there is a rise in the length of the helix around 1.5 angstrom. If there is one amino acid is involved, it will be around 1.5 angstroms. And in a typical alpha helix, the phi values ranges from 113 to 132, and psi values ranges from 123 to 136. So this is the possible relaxation, or the helix can twist. Helix can twist when it is the maximum rotation. If it is going to rotate more than that, so there is some hindrance, or there is a steric hindrance, so hence it cannot rotate, or it cannot twist more than that. So these are the fixed angles for alpha helix. It is 113 to 132 degree, and for phi angle it is 123 to 136 degree. This is very well explained by a noted biochemist, Professor Ramchandran, and his famous Ramchandran plot. The beta sheet, this is also a much simplified structure. Beta plated sheet is composed of two or more straight chains that are hydrogen bonded side by side. So this is one chain, right? So this is one chain, and this is another chain. So you can see the hydrogen of one chain is forming cross hydrogen bonding with the oxygen of another chain. So this kind of hydrogen bondings are going to take place. We get a type of structure called as beta plated sheet, and these beta plated sheets, they can be parallel or they can be anti-parallel. So parallel and anti-parallel means, see, this chain is running in one direction, and this chain is running in another direction. So hence this is an uh, yeah, that's what it is explained. If amino termini are on the same side of each chain, so we have a amino terminal and we have a carboxyl terminal for an amino acid. So if the amino termini are on the same side of each chain, say let us consider, so we have two chains like NH3, assume this is a amino terminal and this is a carboxyl terminal of a amino acid, and again we have one more chain, amino terminal and carboxyl terminal. I am just showing the terminals. Assume that in between it is a linear chain of amino acid. So if both are in one direction, we call it as parallel. So amino carboxyl carboxyl. So this is an example for see, you can see carboxyl 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 amino terminal, you can see. So this is a example for parallel, and here you can see this is an example for anti-parallel. In anti-parallel, so the it will be having amino terminal on one side and carboxyl terminal on the other side, and in the other chain it will be having entirely opposite direction, like 5 prime to 3 prime and 3 prime to 5 prime, right? So hence here it is vice versa, the amino and carboxyl, and carboxyl and amino, it will be like that. Rather amino carboxyl carboxyl, it will not be like that. Okay, so this is the difference between parallel and anti-parallel. So this is a much simpler explanation. You can see here, yeah, this is an simplest representation of beta sheets. So here you can see amino terminal and carboxyl terminal here. So this is amino and this is carboxyl. This is amino and this is carboxyl. It means they are parallel. They are parallel. They are in the same direction. And here we have carboxyl and amino and carboxyl. So hence they are anti-parallel. They are anti-parallel. So beta sheets can be of two types, parallel and anti-parallel. Yeah, these are my references. I hope you have understood the lecture. So secondary structures. So we have four types of conformations. primary structure, secondary structure, tertiary structure, and quaternary structure. The primary structure is a linear structure. The secondary structure has alpha helix and beta sheets. The beta sheets can be parallel or anti-parallel, and the tertiary structure and quaternary structure we will see into much detail uh, into the next lecture. Thank you. Thanks for listening. Stay home, stay safe. God bless you all.